The formation of urine. We begin by looking at glomerular filtration. Glomerular filtration is when we filter fluid from the capillaries of the glomerulus into Bowman's capsule. This is driven by high blood pressure. You've got a higher pressure inside of the glomerulus than you do in other capillaries inside of the body. The higher the pressure, the more filtration that takes place. You filter about 180 liters of filtrate every single day. Now, it should be obvious to you right now that you do not have 180 liters of blood in your body, and therefore we're going to have to reabsorb a lot of this or most of this material back into our bodies. The filtration is going to contain water, of course. It's going to contain different ions, nutrients, it's going to contain proteins, and urea. It is regulated. We can change the diameter of the afferent arteriole. Remember, the afferent arteriole is the blood vessel bringing blood into the glomerulus. If you increase the diameter of the afferent arteriole, you're going to increase the amount of blood that makes its way into the glomerulus, therefore increasing filtration. If you decrease the diameter of the afferent arteriole, you're going to decrease the amount of blood that gets into the glomerulus, therefore decreasing filtration. And it's also regulated a little bit by the sympathetic nervous system. When the sympathetic nervous system sends out an impulse, it causes the afferent and the efferent arterioles to constrict. That results in a decrease in filtration because, quite frankly, if you're having a fight or flight response, it's not necessarily important that you continue to produce large volumes of urine. So taking a look at this picture, you can see that the blood is going into the glomerulus and then it's going to be filtered through the glomerulus and caught by Bowman's capsule. Now, tubular reabsorption is the next process that takes place. And this is the movement of materials from the kidney tubules back into the bloodstream. So whenever we talk about reabsorption, you want to think about materials moving into the bloodstream ultimately. And as we mentioned before, if you're filtering 180 liters of filtrate out of your blood every day, we've got to reabsorb that back into the blood. Most of the reabsorption occurs in the proximal tubule. The reason being is that it's lined with structures called microvilli, and at this point in the course we know that microvilli is there in order to increase surface area for absorption. The glucose that gets filtered, 100% of that gets reabsorbed. You should not have glucose inside of your urine. 100% of the amino acids that are filtered get reabsorbed. 100% of the bicarbonate is going to be reabsorbed. About 99% of the water, so almost all of the water that we filter out is going to get reabsorbed, and of course some of it has to make its way into the urine. 99% of the sodium is going to get reabsorbed. About 50% of the urea actually gets reabsorbed as well. So taking a look at this picture, you can see the tubular reabsorption then is going to be moving from the kidney tubule into the capillaries. This just shows you what the cells of that proximal tubule would look like with the microvilli on their surface so that they can increase surface area for absorption. The mechanism behind this involves a little bit of processing. The sodium is going to be actively transported out of the cells and into the interstitial fluid. Chloride is going to follow. Chloride follows simply due to a charge attraction. So chloride is going to be attracted to the sodium. Now, with a bunch of sodium chloride in the interstitial fluid, water is going to follow as a result of osmosis because remember that water wants to dilute, so it moves to an area of high solute concentration. We would also have some facilitated diffusion of both glucose and amino acids. So as you take a look at this picture, you can see that we are actively transporting sodium out the backside of this tubule, and it's going to make its way then into the bloodstream. Chloride is going to follow via diffusion. Water follows via osmosis, and we can also move other materials via facilitated diffusion. Now, the next process is called tubular secretion. And this is going to be the movement of materials from the bloodstream to the tubules. So we can still take materials that are inside the blood that didn't get filtered in Bowman's capsule, and we can move it into the tubules so that it can be released as urine. Things that move this way are drugs, for example, and remember that I had mentioned before that it's not just, marijuana, but it's also things like Tylenol and Advil. Preservatives will be secreted back into the kidney tubules. Pesticides as well. And things like hydrogen ions and ammonium, and that's going to be there in order to help maintain the pH. Additionally, we can secrete more or less potassium depending on the needs of the body at the time. And then excretion of urine is going to be calculated by looking at what's filtered through the glomerulus, so what is caught by Bowman's capsule. You have to subtract from that what's reabsorbed, so what goes from the tubules back into the capillaries, you subtract that. And then you add to it what was secreted from the bloodstream back into the tubules. That will be what you excrete as urine. So taking a look at this, you can see in picture form now that you take your glomerular filtrate, subtract from it your tubular reabsorption, and then add to it your tubular secretion, and that will be what you excrete as urine. Now, your body, depending on the situation, will produce dilute or concentrated urine. So we're going to begin by looking at how your body creates dilute urine. Or if you don't like that term, you can think about it as excreting excess water from the body. In order for this to take place, we've got to increase our water intake. If we increase our water intake, we know that the water is going to make its way into the bloodstream. If we increase the volume of water inside the bloodstream, it will also increase the blood pressure. As we increase the blood pressure inside of the kidneys, we're going to increase the amount of filtration. Now, the descending limb of the loop of Henle is permeable to water. Since the interstitial fluid around the tubule has a high solute concentration, water will leave that tubule via osmosis. Eventually, it will be picked up by the blood, which I'll talk about in a minute. The remaining filtrate inside the loop of Henle then becomes more concentrated simply because we're taking the water out of that tubule. The ascending limb of the loop of Henle is impermeable to both water and sodium chloride. However, sodium chloride is actively transported into the interstitial fluid. So, as sodium chloride leaves the ascending limb of the loop of Henle, the filtrate, what's left inside the tubule, becomes more dilute, in this case because we're removing the solute from it. Now, the distal tubule and the collecting duct are also impermeable to water without the presence of ADH, which we'll talk about later. And the last thing on here is that urea continues to recycle from the collecting duct, increasing its concentration. So taking a look at this picture, you see first and foremost that there's a color differentiation between the cortex and the medulla. So if you look at that dotted line across this picture, it says isotonic at that level. That means what's inside the tubules and what's outside of the tubules is the same tonicity, has the same solute concentration. As you move down further into the medulla, the interstitial fluid becomes very, very concentrated. And that's important because if you look at the picture now, remember we said that the descending limb of the loop of Henle was permeable to water. As such, water leaves via osmosis. So as we move from the isotonic area at the junction of the cortex and the medulla, move down inside that tubule, you can see that the material or the filtrate inside that tubule gets more and more concentrated by pulling out the water. Now, on the flip side, as we move up the ascending limb, it's impermeable to water, so all the water stays in, and we pump that sodium chloride out. As we move the sodium chloride out of the ascending limb of the loop of Henle, we once again approach an isotonic situation, or the urine is going to become more dilute again. Additionally, if you look at the bottom of the picture, you can see that the urea is recycling from the collecting duct back to the ascending limb of the loop of Henle. As a result of this process, you can see that the volume of urine being released is going to be large and that the amount of solute inside of it will be low, thus giving us our dilute urine. On the flip side, we can create concentrated urine. Now, the process of creating concentrated urine is very similar to producing dilute urine. The difference is when we get to this collecting duct. So, our process of creating concentrated urine is regulated by a hormone called antidiuretic hormone, or ADH. A diuretic is a material or a compound that's going to cause you to lose water. So if we have something called antidiuretic hormone, that would imply that it's going to cause us to conserve water or reabsorb water. ADH is released from the posterior pituitary gland. Its function is to increase the permeability of the collecting duct to water. So the collecting duct used to be impermeable. You wouldn't be able to reabsorb any water out of it. But if ADH is present, you can reabsorb water from the collecting duct. Since the interstitial fluid around the collecting duct is salty, has a high solute concentration, water will move out of the collecting duct via osmosis. The filtrate in the tubules then becomes more concentrated, again because we're losing water or reabsorbing that water back into the bloodstream. So now taking a look at this picture, we've got the same thing that we've talked about before. The descending limb of the loop of Henle would be permeable to water, so as the materials in the tubules move downward, they become concentrated. However, on the ascending limb of the loop of Henle, sodium chloride is pumped out, thus creating more dilute urine. But the catch here is that with ADH present, you can see that the collecting duct now is permeable to water. As such, water is released via osmosis or reabsorbed via osmosis, and the urine is becoming very, very concentrated, so we'll have a low volume of concentrated urine to be released from the body. The process of creating concentrated urine is dependent on something called the countercurrent exchange mechanism. What this is going to do is ensure that the interstitial fluid becomes concentrated as you move deeper into the medulla, thus pulling more water out into the interstitium. The exchange then with the vasa recta, the capillaries around the loop of Henle, pick up the solutes in water, ensuring that the medullary gradient is not disturbed. So this picture shows you that the vasa recta really is superimposed on the loop of Henle, and as water moves out of the descending limb of the loop of Henle, it gets picked up by the vasa recta. That allows us to keep the interstitial fluid around the loop of Henle very, very concentrated or very salty. We need that gradient so that we continue to pull more water out via osmosis.